now is in a training program in the South Florida University. He is responsible for research line that studies the proteomic of tarantula venoms in search of antimicrobial peptide. He is also involved into project in the pathophysiological explanation of the pediatric autoimmune neuropsychiatric disorder associated with streptococcal infection. Please, Dr. Su.
Thank you very much. So, before I begin, I would like to thank both of the institutions involved in this project, the Institute of Biotechnology of UNAM in Mexico City and Anáhuac University, the Faculty of Health Sciences. And to begin, we need to talk a little bit about antibiotic resistance problem that claims over 1.2 million lives per year. And it is estimated by 2050 it will claim 39 million lives. Now, to put in perspective how big this number is, that is the whole population of Canada. And here we have a little example of Acinetobacter baumannii resistance to carbapenems, just to show you that this is a problem that affects worldwide. No one is escaping this, sadly. Now, it's a multifactorial problem, but one of the main issues that we can find, here we have the list of antimicrobial substances approved by the FDA from the 80s to the late 2022. And we can see that there's a slow decline up to 2 medicines approved in 2008 and 2012. And this is really relevant because we can see that we're getting back up there. But if we really take a look into what kind of medicines are they approved, they are all medicines but different routes of administration. For example, metronidazole was discovered in the 60s but was approved orally past year. So nothing new there. So we like to think, like Dr. Carlo Malay said once, most problems that humanity have faced have already been solved by nature. And this is especially true since all living beings had the necessity to fight off bacterial infections in some way or another. Plants, corals, fungi, bacteria themselves, and parasites have created some sort of mechanisms. But also this is true to more complex animals and living beings that produce venom, like insects such as the bees or scorpions, or even snakes that are reptiles. Now, I know some of you may be scared of spiders, so I'm going to put a spider picture. But we are more interested in spiders because they're one of the first animals in all evolution history to develop venom. They are 400 million years old, the first spider ever found, and they have a very specific venom and many proteins. Acanthoscurria gomesiana is a tarantula from Brazil that has a molecule very interesting that kills both gram and negative bacteria. And this is also true for Lycosa polystioma, that is a spider from Europe, and Copeinosalei from Costa Rica. But what's inside of venoms? Well, venoms are very complex. They are actually the most complex liquid in the entire world. But roughly, they're divided into lipids, proteins, amino acids, mucopolysaccharides, and salts. And into proteins, we have two different main kinds, all right? The enzymes, such as phospholipases, that degrade phospholipids, proteases, that degrade proteins, and aggregases that degrade acid hyaluronic, but also low molecular weight proteins. Now, this data is very important. It goes from 3 to 7 kilodaltons, all right? I need you to remember this. And those peptides bind to voltage-dependent calcium and sodium channels. When spiders catch their prey, it gets immobilized, all right? But also, they have antimicrobial peptides. Now, this is what's interesting for us. They belong to the mechanism of innate immunity in many living organisms. And here we have an example of meganin 2, and we can see that it's an antipathic molecule, meaning that it has both hydrophobic and hydrophilic amino acids attached to it. And what does this do? it makes toroidal pores inside of the membrane of bacteria, creating an osmotic disruption, killing it. And luckily for us, there's no no antimicrobial resistance have been found yet against them. And this data is very important for us. 30 to 50% of amino acids are hydrophobic. I will explain later, but for our pH PLC, it's very important to find through the polarity of the molecule where it's found. You'll understand in a minute. So our study aims to evaluate in vitro the antimicrobial effect of the complete venom as well as selected fractions of Poecilotheria regalis venom. What is Poecilotheria regalis? Poecilotheria regalis is an arboreal tarantula from Sri Lanka and India that has a medical importance venom. Most tarantulas don't have a medical importance venom. Usually it's just like a bee sting, but this one causes erythema, joint pain, fever, even vomiting, soft tissue infections by Enterococcus fecalis, and muscle cramps that can last for up to 2 weeks. Now, you can see here the size comparison to a hand, so it's quite a big spider if you imagine it. And this is the one we have in Mexico. Her name given by us is Deborah. And this is the method to get the venom out. I will try to put the video. Okay. So first we put the tarantula inside a container that is hermetically sealed with cotton balls of isoflurane. Isoflurane is a compound that is volatile, so it will make a little gas chamber, so the tarantula needs to breathe it. And here you can see the tarantula is struggling to staying awake. Now, we take once it's asleep, we put it inside a little container so its body is restrained. It needs to be an American Express card, not just kidding. And then a PBS tube is placed under the fangs so we can receive the venom. And saline solution is put on the chelicerae where the glands are, so we can do a little electrostimulation of the venom glands with 7 volts for 20 seconds. It's very hard to see, but you can see like the little drop getting there. And after we get the drop, we finally take it out with a micropipette. Now we're very proud of this technique because most techniques available usually kill the tarantula or harm them. And it took us around a year to figure out how to do it without killing tarantula. And later, we need to do a quantification of the venom. We do a quantification through nanodrop 1 in 280 nanometer wavelength, and then we do a protein analysis to know that everything is okay with our venom. In a CDase page of 20% polyacrylamide gel, 120 volts for 90 minutes. This is the standard that we found out to find the antimicrobial peptides, since they're very small, and usually if we change, they usually don't appear or they fall out. And later, we have to do an antimicrobial evaluation of the total venom. So we tested Escherichia coli and Staphylococcus aureus, ATCCs and BAAs, in Muller-Hinton culture for 24 hours. Now, once we find out that the venom is indeed antimicrobial, we need to do a fractioning of the venom to find which parts of the venom are the responsibles of our results. So we do the fractioning by RP-HPLC. And for those that are not familiar with this, we have a stationary phase, which is a tube, a column, that the venom will go through, and it's made of C18 silica that is non-polar. So non-polar will attire non-polar molecules, and the polar molecules will fall out eventually. Now, as we said earlier, the properties of the antimicrobial peptides allows us to see in the chromatogram a range, and the literature is marked between 30 minutes to 50 minutes of absorption. And later, after we have the the chromatogram done, we have to do the quantification of each fraction in the antimicrobial evaluation of all fractions in the same way that I showed you earlier. Now, our results. First, the venom integrity showed 5 bands of relative molecular weight of 48, 42, 30, 22, and 5 kilodaltons. Now, this is very important. As I mentioned earlier, the antimicrobial peptides go from a range of 3 to 7 kilodaltons. And we can see that the band that had more color was the one that had 5 kilodaltons. So this applies to the concept I mentioned earlier. In the plate diffusion of the complete venom against Escherichia coli and Staphylococcus aureus, we can see 2 graphs. In the x-axis, we have the concentration of the total venom of the tarantula, and in the y-axis, we have the millimeters of inhibition. From Escherichia coli, we can see that we have inhibition from the lowest to the highest concentrations, ranging up to 2 millimeters up to 5 millimeters. However, in Staphylococcus aureus, we only have inhibition in 100 micrograms of total venom of poecilotheria regalis. And this is quite logical, considering that this is a gram-positive bacteria that has a thick cell wall, and we mentioned that the antimicrobial peptides affect the cell membrane that is underneath everything. This is the chromatogram, and as you can see, we had 60 fractions, which is a lot. For venoms, usually they come, tarantula venoms, they come around 50, 40 fractions. 60 fractions was pretty good number. And we could see that going through 30 to 50, as I mentioned earlier, there's most of our fractions. So this was very promising to us. And while we tested the fractions, we could see that there was 21 fractions that were antimicrobial against Escherichia coli, whereas 18 fractions were antimicrobial against Staphylococcus aureus. To give a perspective, Mexico has the second largest species count of tarantulas in the world, and we have tested many of them. And usually we find 4, 6 fractions that are antimicrobial in tarantulas from Mexico. So finding 21 fractions was really exciting for us. And we could see that in Staphylococcus aureus, all fractions were present also in Escherichia coli, but not vice versa. All right? And the fraction 8 and 31 were the fractions that had the highest inhibition in both bacteria. And we need to, in the future, analyze these fractions with more detail so we can know exactly what is going on inside of them. However, we also saw that the controls we used, that was penicillin and ciprofloxacin, were still higher than all of our fractions in our experiment. So we were the first to find gram-positive and gram-negative antimicrobial activity in large percent of venom fractions in an Asian tarantula. And for the future, the fractions 31 and 8 need to be processed in mass spectrophotometry to know the amino acid composition to then assess minimum inhibitory concentration against non-resistant and resistant bacteria in vitro. With this data, we could possibly in the future help not only to battle with antibiotics, bacterial infection, but also reduce antibiotic resistance. And I want to thank and present to you all my team that we work with in Mexico City. And if you have any questions, I'm here to help. Thank you very much for your attention.
Thanks a lot. A question by me. The final target of the toxin is?
The toxin.
The target of the toxin for the antimicrobial effects. is against the peptidoglycan
Oh,
or other.
it goes against the cell membrane.
Yeah, the cell
But
membrane.
we could think, well, we have cell membrane as well, so how can we access that this is antimicrobial and it's not cytotoxic? Well, antimicrobial peptides have the property of going against lipoproteins, lipopolysaccharidae that are on the membrane of bacteria. So it's specific against them, specific against the cell membrane of the bacteria.
Okay, so no effect on the membrane of the cells,
Well,
the human cells.
we haven't tried it out.
Okay.
We need to try it out, but
Yes.
in theory.
In theory,
In
not.
theory, yes, of course.
Yes, this one is
Yes.
important.
Yes.
Okay, thanks. Do you have some question? Yes, one.
Since your venom fraction is a peptide, if I'm not wrong, so what about the penetration and absorption? So
Okay.
One of the most important problems using the peptide as an antimicrobial agent is absorptions and also the penetrations problems. So how can you deal with or comment on this?
Yes, well, when we haven't even tried cell culture trials yet, we are just trying to find the peptides, but we have already thought about it since there's, I don't remember the name of the, in English, but to protect the peptides, there's a way to protect.
Do you mean the nanotechnologies?
Yes, exactly.
Even right now, the nanotechnologies to wrap up the peptides, only 15% of absorption is the maximum dose that you can do. 15% is really low.
Yeah, well, we haven't figured out that step yet. We need to first understand the properties of the peptide we're finding, because perhaps we're thinking, oh, it could help infections in soft tissues, but at the end of the day, it has better absorption in other tissues, ophthalmic volume or oral intake. We do not know yet. We should try. We should make more trials first to understand if it's cytotoxic or not. Theoretically, it isn't, but we need to know more. And later to assess, of course, how do we do the biotransfusion of the peptides into the tissues. Yes.
A question more, curiosity. In nature, tarantula is a resistant tarantula to bacterial infection.
Yes.
Yes, for
yes.
this reason
yes.
you
That's-
decided.
Yes, the hypothesis started there because most tarantulas eat very dirty animals.
Yes.
They eat roaches, they eat rats, they eat everything, and they never fall sick of bacterial infections. Whereas parasites, they get always killed by them, but parasites are way more complex to kill, sadly. But when we started to see, because I've been working with tarantulas since I was 10 years old.
Oh.
Yes, I really like them.
Do you like
And-
tarantula?
Yes.
Okay.
And I've never seen one of them get sick ever.
Yeah.
And for me that was very interesting to think, well, what's going on inside of them? What is the mechanism that protects them? And inside a French book that I once found, it mentioned that the venom was not only to eat and to kill, but also to preserve its life. And it's quite logical since they have been making this venom for 400 million years. So it's a very slow evolution problem that has many proteins that we don't even know.
Yes, but the animal toxin is a very exciting area of research from spider, for tarantula, for snake,
Yes.
of course. So
Yes, the field in
it's a
tarantula.
nature project.
Yes.
Nature.
The field in tarantula is quite new and is
Yes.
quite unexplored since, as I mentioned earlier, it's very hard to get the venom. And it's not very profitable to kill your tarantula every time you get the venom from it. It would be very
Of course.
complex and unethical, of course. So,
Yes, the one problem with the toxin with the snake,
yes,
and to prepare the antitoxin.
yes,
And the European Union said that is not ethical
yes,
to take the venom for the snake.
yeah,
And so now in Europe you don't have toxin and
yeah,
drugs to combat
the
the
bites.
snake.
Yes, of
for the
course.
bites.
Yeah, and since this is a really fairly rare spider, the bite reports are quite rare, so that also pushes back research, since it's not as common and the needs to study its venom is not as important as a snake that kills 1,000 people per year.
Yes, I know.
Yes.
This one is what the European Union decided. So if you are bites by a viper, you don't have the toxin in the European Union.
Yes.
No more.
Yes.
Okay, thanks a lot.
Thank you. Thank you very much.